Thank you very much. Welcome to my presentation. My name is Benjamin Mooney, and I'm here to present my thesis, Carbon Flux in a Northern Norwegian Fjord, the potential contribution of the small copepod, Microsetella norvegica. So as the title suggests, my study is focused on Microsetella norvegica, which is on the left of the screen here. From now on, I'll just refer to it as Microsetella. It's a small pelagic harpacticoid copepod, and it's widely distributed in coastal ecosystems around the world, where it's often cited as being numerically dominant. It's known to eat phytoplankton, and it also serves as an important fish food source for many fish species. And interestingly, and vital for this study, it's also known to eat marine snow. The problem is we don't know when, where, or how much marine snow Microsetella consumes. So we don't know the impact it has on breaking down organic material. But let's start with what is marine snow and why is it important? And to explore this question, we can talk about the formation of marine snow. So organic material, which is represented by these small green shapes here, exists in the euphotic or the photosynthetic zone. And it exists in the form as phytoplankton, which eventually dies, fecal pellets, and mucus, as well as other less contributing components. But this material begins to fall out of the surface water. And as it does so, it can begin to stick together. And these aggregates of organic material are what we know as marine snow. Now, as they sink, they're broken down and they're remineralized by different groups such as bacteria, microorganisms, and copepods. And this reintroduces the organic material back into the ecosystem as higher trophic levels eat them. Not all marine snow is consumed or remineralized, and those that isn't sinks to the benthos or the seafloor. And here it can become trapped and it can be sequestered for a very long time. And this is one of the ocean's #1 ecosystem services. It removes carbon from the atmosphere, and it therefore helps to control global climate. So it's important we know how much snow is remineralized before it reaches the seafloor. Small copepods in previous literature have thought to not be so, or they don't contribute much to organic degradation. But as a numerically dominant species among secondary producers, and the fact that it consumes marine snow, it's important that we understand exactly what influence Microsetella has on aggregate degradation. With this in mind, the three aims to this study are to, first of all, determine the seasonal association of Microsetella with marine snow. Secondly, we want to understand if and how the presence of phytoplankton affects this association. And finally, we want to estimate the seasonal abundance of Microsetella norvegica. So in order to investigate Microsetella's association with marine snow, we need to know where these copepods are in the water column. We also need to know where marine snow is positioned. So traditionally, zooplankton, and so copepods, are sampled with WP2 nets, and you can see one of these on the left of the screen here. These are dragged vertically through the water column from depth to the surface or to shallower water, and they typically have a mesh size of 180 micrometers. And actually, it's been shown that a mesh size of 180 micrometers is shown to undersample copepods with a body size of less than 800 micrometers. Microsetella's maximum body length is 550 micrometers, and it's quite a slender body. So historically, studies using these nets have underestimated the abundance of Microsetella. And maybe more importantly for this study, they're not able to investigate fine-scale distributions, so exactly which depth we find the individuals at. So in order to identify the exact depths Microsetella are found, and to have accurate abundance data, we used a video plankton recorder. And that's what you can see on the right of this screen. The video plankton recorder, or the VPR, is an optical sampling tool. It's lowered below a stationary vessel through the water column, taking high-speed photos, about 21 frames a second. Now, these images are tied with the exact depth they're taken. So as we count individuals on a frame, we can see exactly what depth it's associated with. Samples for this project were taken on cruises which occurred through the years 2013 and 2014, and you can see the months highlighted on the timeline below. We felt like these months gave us a good spread throughout the year during important productive and non-productive seasons. So just to show you the physical characteristics that were taken with a CTD attached to the VPR, we're looking at temperature and salinity here. The sampling periods are separated by color, and the main thing to take away here is that, as predicted in the summer months, May and August, the surface water is warmer, and the water is generally well mixed, with the exception of October, which has strong stratification at around 45 meters, and August, which is where we see the largest difference in salinity and temperature between the surface and the bottom. So the study site. The data for this study were collected in the inner part of Porsanger fjord, which is a fjord in the northernmost part of Norway. The inner part of this fjord is known as Osterbottom, and it has extremely low temperatures compared to other parts of Porsanger fjord and also compared to other Norwegian fjords. In fact, it gets so cold that it reaches sub-zero temperatures. The water reaches sub-zero temperatures in the winter. You can see the remains of some sea ice, in a photo I took in mid-April this year. So it gets seriously cold. And the area is unique in the sense that it's truly an Arctic ecosystem in a subarctic latitude. There have been polar cod caught at Osterbottom historically, which is a genuinely Arctic cryopelagic species. So the area is exciting and undergoes huge changes from summer to winter, and it even supports its own fish stocks. So let's start looking at some results here. Again, each sampling period is separated by color. You can see that on the legend on the right. On the left-hand side graph, we can see Microsetella's depth distribution, looking at individuals per meter cubed. The numbers on the right of this graph represent the weighted mean depth. Now, I chose to use the weighted mean instead of the peak abundance depth because Microsetella are living organisms. And I think as such, we have to consider each individual as trying to position itself at the optimum depth. We can see that Microsetella are indeed very abundant. The highest abundance was in October in blue, and we can see at its peak there's over 200,000 individuals per meter cubed, which is a lot. There's also quite high abundance in the summer months. If we look at the graph on the right, we're looking at marine snow, and we're looking at marine snow particles per meter cubed here, so density, if you like, versus depth. And this data has been square rooted for the sake of visualization because we just saw so many more particles in May. The numbers on the right of this graph represent the highest abundance, or the peak density, if you like. And if we draw lines from the peak density of marine snow over to the Microsetella graph, we can see that in the summer months, May and August, the peak abundance or density of marine snow depths, they're actually below the weighted mean depth of Microsetella. But if we do the same thing for the winter months, October and November, we can see that the peak density of marine snow actually coincides quite well with the weighted mean depth of Microsetella. So the question now is, why do we see differences between the summer and the winter months? Well, to investigate this, we can look at each month individually. So here are the results from May. The top graph shows fluorescence and Microsetella abundance on the x-axis, and of course depth on the y. It's important to note now that fluorescence, of course, implies the presence of phytoplankton. So the higher the fluorescence, the more phytoplankton we have in the water. And we can see quite high fluorescence, over 6 micrograms per litre. So we're in the middle of a bloom. And we know we're late on in the bloom because we have a subsurface fluorescence maxima there. And if we look at this graph, we can see that the distribution of Microsetella seems to fairly well match the distribution of fluorescence. If we go down to the bottom graph, now we can see marine snow versus Microsetella abundance. And we although we have a high density of marine snow here, we don't see them distributed at the same depth. So in an attempt to quantify this, I did some Spearman correlation coefficients. This essentially investigates how well the relationship can be described between two variables, ranked variables, importantly. The way to interpret this matrix quickly is that the larger and darker the circles, the stronger the correlation. Blue circles are positive correlations, red circles are negative correlations. So that's why if we look at marine snow versus marine snow, for example, we have a very dark, large circle, a perfectly positive correlation. So if we look at Microsetella versus fluorescence in the top right, we have a large, fairly dark circle. So it's a strong correlation. And I've put the p-values on top here. and I had a threshold of 0.05. So fluorescence and Microsetella seem to be have a good relationship, and it's not due to chance. It's significant. Marine snow, on the other hand, and Microsetella, not a strong correlation. Positive, but not strong, and actually not significant. So it could just be down to chance. Okay, let's look at May. And again, strong fluorescence values here, over 6 micrograms per liter. And the distribution of Microsetella does seem to match the distribution of fluorescence again. It's quite convincing, actually. If we look at the bottom graph, we don't see Microsetella and marine snow distributed at the same depths, at least not in their highest abundance. So we can again quantify this looking at a Spearman's correlation coefficient. And yes, again, fluorescence, a strong positive correlation, again, significant. Marine snow, although the relationship is significant, it's a negative correlation, and it's not so strong. On to the winter months, and we can look at October. So here we start to see something quite different. If we look at the top graph again, Microsetella's distribution is totally not coinciding with the distribution of the phytoplankton or fluorescence in the water. But if we look at the bottom graph, there does seem to be a trend. As the abundance of marine snow increases, as does Microsetella's abundance. And so if we just quantify this, a strongly negative correlation with phytoplankton or fluorescence, as we would expect, and a positive correlation with marine snow. Not that strong, but it's a significant correlation with marine snow, and with fluorescence is non-significant. So we can begin to ask ourselves why. And if we look at the strength of fluorescence here, we begin to reveal some possible answers. So the highest fluorescence value here, below 45 meters, it's less than 0.6 micrograms per liter. So there's not much phytoplankton in the water. So let's look at November, and now we see almost no fluorescence, extremely low values, so we don't really have any phytoplankton in the water. If we look at the bottom graph, there's quite convincing similarities between the distribution of marine snow and Microsetella. And again, now we have a not strong correlation with fluorescence. In fact, it's negative, and we have a strong, significant positive correlation with marine snow. So let's go back to our aims. We wanted to investigate the seasonal association of Microsetella with marine snow. Yeah, we've done this, and it looks like Microsetella are associating themselves with the same depths as marine snow during non-bloom periods. And this helps us to understand the second point, to understand if and how the presence of phytoplankton affects this association. There seems to be a preference with phytoplankton in the summer months. And finally, we estimated the seasonal abundance of Microsetella norvegica, and actually the abundance is higher than reported in previous studies using these WP2 nets with the mesh size of 180 micrometers. So to wrap up, there's a clear seasonal variation in both abundance and distribution of Microsetella. The distribution of Microsetella appears to be driven by food availability. So in the summer months, during bloom periods when we have lots of phytoplankton, Microsetella is distributed in shallow water in the same depths as phytoplankton. In the winter months, in absence of phytoplankton, Microsetella are associating themselves with deeper depths where we find marine snow in high abundance. So, as I mentioned before, the contribution of small copepods to organic degradation has previously been underestimated. But considering the high abundance of Microsetella reported in this study, combined with the fact that we know they degrade marine snow and they're associated at the same depths of marine snow in the winter, it seems quite obvious that there's a high influence on the vertical flux of marine snow. So this is hugely important because as our oceans change, our ecosystems change too. Communities and food webs will be altered. And as we move forward, we need to prioritize investigating the contribution that other dominant groups have on the degradation of marine snow. So as we can model community change, we can also predict how carbon sequestration will proceed with time. Thank you very much for listening to my presentation.